One is I've got note cards for you that I'll hand out at the end of the period, and you'll need to come up and get one from me individually. And that's one thing. As I noted last time, you will have to bring a note card to the final. So if you don't bring a note card that I gave you to the final, you will lose points. So make sure you bring a note card that I gave you to the final. And yes, I have a way of knowing. Okay? Oh, evil, right? That's number one. Number two, when are you going to post those grades, Kevin? I'm hoping to get, and I'm hoping I will post grades this weekend, so you'll know where you are with stuff. If you have any questions, of course, let me know. And what I will do is I will post the grades based on your cumulative score to this point. So that will include the 2 exams that I've given you and the best of the 2 exams that Dr. McFadden gave you. Okay? So that's what the grade distribution will be based upon, so you'll see where you stand in the class with respect to that. Yes? It'll be posted on the web on the class schedule page. Okay? Third, Kevin, are you going to do a review session? Yes. Kevin, when's the review session going to be? I don't know. So I haven't been able to get a room scheduled yet. I will get a room scheduled. And when I do, I will post that on the class schedule page. It will not occur this weekend. Okay? So the review session will not be this weekend. It'll be an evening sometime next week. I'm guessing Monday, but I've got to also look at my calendar to get that set up as well. Okay? But I will post that on the web page, and I'll send an email to the class as well. We all clear on that? Okay. So as I said, I have a surprise also today for you. So be alert. You never know when you might benefit from that surprise. So that said, we've got a lot to cover. So let's talk. Let's talk about continuing to talk about nitrogen metabolism. And last time when I finished, I had really gotten to the relationship of these things to the citric acid cycle. As I said, I don't expect you're going to memorize anything like this, but I want you to really, the thing I want you to take away from this is the fact that we like to think of metabolism as very compartmentalized. Here's glycolysis. Here's the citric acid cycle. Here's the pentose phosphate pathway. Here's the Calvin cycle. But these are all inventions of man. These are just little boxes that we drew around. In most cases, these reactions are all happening in the same place. This is a really good example of the fact that these pathways are interconnected. And even though the citric acid cycle is happening in the mitochondrion and some of these other things are occurring out in the cytoplasm, molecules can move by shuttles across membranes to experience these sorts of things that we're talking about. So the bottom line is that amino acid metabolism is intimately tied to cellular energy metabolism, intimately tied to it. All right? We can derive energy from amino acids, and we can derive amino acids from energy-containing molecules. Very important. Okay? I mentioned last time the fact that there are essential amino acids, which are amino acids that we can't synthesize, that we have to have in the diet, just like we had essential fatty acids that are fatty acids that we can't synthesize that we have to have in the diet. Okay. And I'll actually briefly mention those in just a little bit. Okay. So the bottom line is that there's a lot of things that can interplay here. What we see is that most of the amino acids, in one way or another, can interact with citric acid cycle. Most amino acids in one way or another, not all, but most, as we will see, can interact and be part of the citric acid cycle. At the very least, they can be a part of this guy over here, the acetyl-CoA. So if we take acetyl-CoA as part of the citric acid cycle, we're really talking about a good chunk of the amino acids that are involved in that. Okay. What I would like to do right now is to turn our attention, well, actually, let me back up on that. I'm going to skip over a few things here, but I want to turn our attention to essential fatty acids since I've already mentioned those essential fatty acids. All right? Essential fatty acids we have to have in our diet because we don't synthesize, or we don't synthesize very well. In some cases, like you can see here, mammals synthesize arginine but cleave most of it to urea. Essential for children, but not essential for adults. Meaning that we synthesize some, but not enough as an adult. So some of these have little asterisks by them indicating that, our needs aren't always what we're able to synthesize. When we see non-essential, these are ones that we can synthesize. And as I indicated to you last time, these are, in many cases, frequently very easy for us to synthesize. Aspartic acid, of course, coming from oxaloacetate. Glutamic acid coming from alpha-ketoglutarate. And alanine coming from pyruvate. In each case, that was a simple transamination reaction that gave us that amino acid, and they're very easy to make. Over here, we see things like the amino acids that have benzene rings. Benzene rings are not easy to synthesize. Phenylalanine, tryptophan. Okay? Tyrosine is over here, and you say, well, that's got a benzene ring. We can make tyrosine. Well, it turns out we make tyrosine from phenylalanine. So if we have phenylalanine in the diet, all we have to do is put a hydroxyl group on it, and we've got tyrosine. So these amino acids, and if you count, of course, you'll see quite readily that there's about 10 in each group. And no, I don't expect you're going to memorize that. Just the nutrition majors need to know that. Okay? So on the exam, you'll just put down a little box, X box, and it says nutrition major, and then there'll be a little question. Which are the essential? I would not be very popular if I did that. Okay. Now I'm going to come back and I'm going to talk. I'm jumping around a little bit today. I recognize this. When we look at the breakdown of amino acids, we put them into really two categories, but really three. Okay. So the two categories that we put them into are whether they are what we call ketogenic or glucogenic. Okay. Ketogenic or glucogenic. Glucogenic amino acids are those that can be made into intermediates that can be made into glucose. Ketogenic are those that are made only so these over here are only into glucose synthesis intermediates. These are only into ketone body synthesis intermediates. And these on the right can contribute to both. Well, we see that most of these guys here contribute to gluconeogenesis intermediates because, for example, oxaloacetate is a gluconeogenesis intermediate. Okay? So most of the amino acids that we have in their breakdown are glucogenic, meaning that they can be made into intermediates that can be made into glucose. Okay? Now, what I wanted to show you was a little bit about why or how it is that ammonia and amines are handled in our body. And it's very important to understand that amines, more specifically ammonia, is a toxic substance. Our body has to be in what I like to describe as nitrogen balance. If we're out of nitrogen balance, that either means we've got too little amines or we've got too many amines. And for our diets, that generally means we've got too much in the way of amines. Our body has a way of balancing that, and the way that our body balances amines is via the urea cycle. So the urea cycle is another cyclic pathway. And no, we're not going to go through the individual reactions of the pathway. But there's some things about the pathway that I want you to understand. Okay? One of the main functions of the pathway is to make this compound urea. Okay? I actually have a song I was going to do, but it's kind of a boring song, so I didn't. I decided against playing it today. It's called. They call the stuff urea. Anybody know the song? They call the wind Mariah. Yeah, some people do. Okay. So anyway, but it's not a real exciting song, so I decided not to play that. But anyway, they call the stuff urea is important because urea is a way for us in our body to get rid of excess nitrogen. And we get rid of it in our urine. Okay? It's one of the things that helps contribute to the smell of urine. Okay? And if you look at the structure of urea, you see it's very efficient at taking away amines. There's an amine right there, and there's an amine over here. If you look at the urea cycle, what you will see is that it's tied to things like arginine. Arginine is an amino acid that's full of nitrogens. Okay? Look at arginine. 1 nitrogen, 2 nitrogens, 3 nitrogens. Okay? Here's aspartic acid coming in, bringing in a nitrogen. Okay? What else do we have? We have fumarate, which is splitting out and going to a citric acid cycle intermediate. All right, I'm sorry, going out to be a citric acid cycle intermediate. Okay. Now, this cycle occurs mostly in the cytoplasm, but there are a couple of reactions that occur in the mitochondria. What that means, therefore, is that there has to be a shuttle system that moves things into the mitochondria, and there also has to be a shuttle system that moves things out. Okay? This cycle includes 2 unusual amino acids. And these 2 unusual amino acids, all right, are not found in proteins. So sometimes I get asked the question, are there amino acids in the body that are not found in proteins? The answer is yes, there are. And these 2 are very common, ornithine and citrulline. Okay? Some of the pep drinks that people think give them pep, which, by the way, I think people should avoid like the plague. I'm guessing the nutrition majors will agree with me on this, are full of citrulline. What's citrulline giving you? Here's citrulline right here. Citrulline is giving you fumarate. And what's fumarate giving you? Intermediates in the citric acid cycle. Does it give you energy? I can't tell you. Does it make you smarter, which is the other things people seem to think about citrulline? I don't know. I never knew anybody who was a citrulline addict who was overly intelligent. So, dumb joke. Okay. All right. So this is a cycle. And every time the cycle turns, urea comes off. Every time the cycle turns, urea is kicked off. So the more excess amine that we have, the more the more the urea cycle is going to run, because it's a way for us to excrete nitrogen. Mammals excrete urea as their way of getting rid of excess nitrogen, with one exception, and the one exception is Dalmatian dogs. Anybody have a Dalmatian dog? Nobody has a Dalmatian dog. Okay. Dalmatian dogs are unusual. They don't excrete urea. They actually excrete their nitrogen in the form of uric acid, which, as we will see, causes some severe problems for those dogs. Okay? So uric acid is a problem, as we will see. Birds excrete uric acid. Okay? Birds excrete uric acid. Bird poop is full of uric acid. All right. Now, let's see. What else I want to say? Let's talk about uric acid. Why not? Okay. All right. I'm going to talk in a little bit about the metabolism of nucleotides. The metabolism of nucleotides. That's the last metabolism we're going to talk about, is the metabolism of nucleotides. And it turns out that the metabolism of nucleotides, both the synthesis and the degradation, is interesting. I say that because I'm a biochemist and I have to say that. All right? But the breakdown, particularly of purines, is of interest to us right now. So purines include adenine and guanine. The adenine-containing nucleotides and the guanine-containing nucleotides. When they break down and create uric acid. The breakdown products of purines create uric acid. Now, normally uric acid, as we're following it in our body, will go and get broken down into allantoin, allantoic acid, blah, blah, and finally down to urea. Okay? That goes out in our urine and we're done with it. If this cycle here gets interrupted in some way, by perhaps we're overloading it or perhaps an enzyme isn't working very well, what will happen is uric acid will accumulate. We are making uric acid. Just like birds are making uric acid, but we're not using it as a way of excreting nitrogen in our urine. It turns out that you can't really excrete uric acid very well in your urine. It's one of the reasons we don't do it. And the reason is because uric acid turns out to be fairly water insoluble. Fairly water insoluble. Okay? Now that, it turns out to be a real problem. People who make too much uric acid will, in some cases, develop an excruciating pain in their big toe. Anybody know where I'm headed with this? Gout. Okay. So gout arises from uric acid crystallizing, and it crystallizes in the lowest part of your body. The lowest part of your body, in this case, being your big toe. That's where it accumulates. And for people who get this, it's excruciatingly painful. Okay? Gout turns out to be, and it's sort of in the old silent talkie movies, gout was always considered this hilarious disease where this guy is holding his big toe and he's running around and he's in pain. But I'm sure if you've ever had this pain, it's not a very good pain to experience. It turns out there appears there might be benefits to having gout. And everybody's going, boy, he's really crazy. Anybody know where this is headed? Anybody? If you do, raise your hand. Anybody heard my lecture about this? Nobody's heard my lecture about this. I've got a surprise for you if you do. What's that? Nope? Okay, that's fine. All right, so it turns out that there appears to be an inverse correlation between incidence of gout and occurrence of multiple sclerosis. Okay? People who have gout tend to have a lower incidence of multiple sclerosis than those who don't. Right? That's kind of an interesting observation. It's not really known why that's the case, and it's not a giant relationship, but there is that relationship between the two. Now, the reason that Dalmatians have such problems is because they're making a lot of uric acid, and uric acid, because it causes pain in your big toe, can accumulate in joints. It can cause all kinds of problems. So Dalmatians, frequently, people who own Dalmatians will say, wow, I'm taking this dog to the vet all the time. It just doesn't seem right, et cetera, et cetera. And it's because their body's just really full of uric acid, and it makes the joints very sore and tender and so forth. Okay. All right. Well, let's talk briefly about the metabolism of the nucleotides, and then I can promise you we will stop. How's that? When we look at the structure of nucleotides, at one level they seem like they're big and hairy. Right? This is the general structure of a purine. It's got two rings. The rings have carbons, they have nitrogens, etc. But if we look where those carbons and nitrogens come from, we discover that for the most part, they come from fairly simple things. The central part of it, comes from glycine. Here's a nitrogen that comes from aspartate. Here's a carbon that comes from carbon dioxide. Here's a nitrogen that comes from glutamine. Here's another nitrogen that comes from glutamine. And the only things on here that have big mouthful names are actually fairly common things in our body that donate single carbons. Okay? Folate, that's a typo there. Okay? Folates, everybody heard of folates? What are folates important for? For what? Spinal bifida, okay? First prize. Brand new CD, Metabolic Melodies. It was just released this past week. Okay, so people who answer my questions will get, I've got one more, will get that golden prize. Okay, hot off the presses. So that's what folates are important for. Okay, they put folates now in breads and cereals because pregnant mothers who are deficient in folates have infants that tend to have a higher incidence of spinal bifida. Okay, so that's an important consideration. Anyway, so as I show you this figure, you see that even though the base in a nucleotide looks very complicated, the base itself is made from very simple components. When we think about the evolution of life on Earth and the chemical origins of life on Earth, if we think about the fact that we can make nucleotides and the bases in nucleotides from very simple compounds, we begin to think that if we look on places and other worlds where there are these simple compounds that are there, we might very well find life forms that have a basis like what we have. Okay? And so that's part of my reason for showing you this. Okay, Well, when we look at the synthesis of these guys, okay, it turns out that the synthesis, and I'm showing you here the overall synthesis, and I've skipped a whole bunch of steps in getting to this point. But if we look at the overall synthesis of these guys, we're following the synthesis of purines. These are things that will ultimately become AMP and GMP. Okay? The synthesis of both AMP and GMP proceeds along the same lines. They start with the same things, they go and they get to the same point, and they finally make this molecule here called IMP. Anybody know where you've seen IMP earlier this term? There's a CD waiting on it. IMP. Anybody know where you've seen the nucleotide or the base I? You've seen it. Wobble base. You've already got one. I'm going to give it to you. The wobble base in a transfer RNA. There you go. Enjoy. Okay? The wobble base in a transfer RNA was I. That was because it could pair, remember, with multiple things, right? Okay. The I was there. Well, the I turns out to be a branch point in the synthesis of AMP and GMP. One of the things we see about nucleotide metabolism that's really interesting is the fact that nucleotides metabolism is more tightly regulated than any other metabolic pathway. You thought, oh my God, glycogen regulation was more than I want to think about. If this is even more than that, he's really going to get us in this last half hour, right? Well, in fact, what matters with nucleotide metabolism, the reason that it's so tightly regulated is because the balance of nucleotides is critical for the cell. If cells get too much or too little of one of the nucleotides, the cell will be very prone to mutation. And mutation, as you know, leads us, as higher organisms, ultimately to cancer. So cells have to be very, very careful not to make too much or too little of any given nucleotide. Very important consideration. All right? Well, I'm saying that. Why am I telling you this? By the way, I'm skipping a lot of the regulation. I'm just showing you some very simple things about the regulation. Suffice it to say, there are many more things besides what I'm telling you here. One of the things I want to tell you here, though, is actually shown on this figure. All right? If we look at the synthesis of AMP, we look over here and we say, well, this guy doesn't have a phosphate. Where does the phosphate come from? It comes from GTP. And if we look over here at GMP and we start with this guy that doesn't have a phosphate and we put phosphate onto it, where does that phosphate come from? It comes from ATP. This is one of the ways that cells balance. If cells have too little GMP, they have too much ATP, and ATP is donating phosphate to make this guy happen. Conversely, if cells have too little AMP and too much GTP, they go this way to make this reaction happen. Balance being very important. Okay? Now, as I said, there are many ways that that balance occurs. This is one. Another way that balance occurs here is that the enzyme, and by the way, you don't need to know the names of these enzymes, but these enzymes that you see here, this enzyme is inhibited by the end product, which is AMP. And this enzyme is inhibited by the end product, which is GMP. The bottom line, feedback inhibition is also ensuring that the cell doesn't make too much of any given nucleotide. Feedback inhibition is making sure that the cell doesn't make too much of any given nucleotide. So what we're seeing here is how the purines are regulated. Okay? I'm going to turn our attention. I'm going to skip to the pyrimidines, and then I'll come back to the breakdown. So this is how we make them. All right? So there's the overall making pathway. No, you don't want to see that. Okay. All right. If we look at the synthesis, and now you're seeing a lot of the individual reactions for the synthesis of the pyrimidines, and again, you don't need to know the intermediates. We look at the synthesis of the pyrimidines, you can actually see how simple the compounds are that we're starting to make pyrimidines. Remember, pyrimidines only have a single ring. The bases and the pyrimidines only have a single ring. The bases in pyrimidines include uracil, cytosine, and thymine. Okay? There's bicarbonate. There's the amino acid glutamine. There's ATP, and there's water, and we make this molecule. Okay? Did Dr. McFadden talk about ATCAs? No? Okay, so I'm going to tell you just for a minute about ATCAs. ATCAs is commonly described when people talk about how enzymes work, because it's the classic enzyme that is used to describe feedback inhibition. So I'm going to, if you didn't get feedback inhibition earlier, I'm going to flesh it out for you here. Okay? ATCAs catalyzes one of the early reactions in the synthesis of pyrimidines. One of the early reactions in the synthesis of pyrimidines. Okay? We don't really even have to worry too much about what the reaction is that it catalyzes. But it turns out that ATCAs is the enzyme in pyrimidine biosynthesis that is regulated. And it's regulated in an interesting fashion. It's regulated by the end product of the pathway. Well, the total end product of the pathway isn't seen here. There's UMP, that's uridine monophosphate. If I put an additional phosphate on there, I've got uridine diphosphate. I put an additional phosphate on there, I've got uridine triphosphate. Okay? Uridine triphosphate, all right, can be made into cytidine triphosphate. So now you can see how we make U's and C's. All right? It turns out that CTP is the end product of this pathway. CTP. CTP inhibits this enzyme. It inhibits ATCAs. So the end product of the pathway is saying, once CTP starts to accumulate, this means the cell has too much CTP, and since UTP was used to make CTP, the cell also has too much UTP. Okay? So the end product of the pathway is shutting off the entire pathway so that we don't have too much of the pyrimidine nucleotides. Balance is important. Okay? Balance is very important. If all that we did was turn this enzyme off, that would be one story, but it turns out this enzyme can be turned on by something else that's equally important. It's turned on by ATP. So the enzyme is turned off by the end product of the pathway, CTP. It's turned on by ATP. Now, why is that important? That's important because of balance. Too many purines turn on pyrimidines. Think about this. The bases in a nucleic acid. A pairs with T. A is a purine. T is a pyrimidine. G pairs with C. G is a purine. C is a pyrimidine. If I have too many purines, it makes sense to boost the number of pyrimidines. If too many purines, it makes sense to boost the number of pyrimidines. And that's what's happening. So ATCAs is contributing, excuse me, to the balance between purines and pyrimidines and making sure the cell doesn't make too much in the way of pyrimidines. Okay. Now, I haven't said anything about T yet. T is made in a totally different way, and we probably will not go through that. Okay. Questions about what I've said? I'm running on here. Question? Yeah, it's a good question. So can the pyrimidines turn on the purines? It turns out they don't need to, because this helps to balance both. If I have too much in the way of pyrimidines, this shuts off pyrimidines. And if I have too little in the way of pyrimidines, then the ATP can turn it on. So that one enzyme actually helps to balance both of those. Pretty cool. You guys ready for a joke? Okay. Anything to stop talking about biochemistry, right? Okay, this is a dumb joke, all right? People used to tell ethnic jokes and sexist jokes, and I don't believe in telling those. So, but the one that they still let people tell are dean's jokes. So I'm going to tell a joke about a dean. Okay. So there's two deans sitting on an airplane, right? And they're flying from New York to Los Angeles. Okay. And the pilot comes on, and the two deans are sitting there, and the pilot says, "Have you heard this?" "No." "You've heard this?" "No." "Oh, the tie's cool." "Oh. Don't you think he looks just like me? Don't you think he just thinks just like me? Or more likely you think maybe his hair is just like my hair." Okay. Two deans sitting on the thing, and the pilot comes on, and he says, "This is your pilot speaking. We're flying from New York to Los Angeles, and we'll be flying, a Boeing 737, blah, 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 and expect to arrive in Los Angeles at 11 p.m. Two deans sit down, they put their trays down, they get their food and all this and that sort of stuff, right? And they're riding there for a while, and all of a sudden they hear this boom! What the hell was that? Pilot comes on and goes, "Oh," he says, "awfully sorry about that." He says, "Looks like our number 4 engine has just conked out." But he says, "A Boeing 737 has 3 engines, and the only effect it's going to have on our flight is we're going to be about an hour late. We'll be arriving in Los Angeles about midnight." And Dean says, "Okay, we'll get there, right?" So they're sitting there, and they're getting ready, and they're watching the movie, and all of a sudden, boom! Right? The pilot goes, "Wow!" He says, "Sorry, folks, I've never seen this happen before, but our number 3 engine has just conked out. Our flight's going to be delayed a little bit getting into Los Angeles. We're only going to be arriving there at about 1 a.m. Dean say, "Okay, well, we'll get there, right?" They're just about to nod off to sleep, and all of a sudden, boom! The pilot says, "Ladies and gentlemen, I've never been on a plane where I've ever seen this happen before, but the number 2 engine has just conked out." He says, "Number 1 engine, get us into Los Angeles at 2 a.m. One dean looks at the other and he says, "If that last engine goes out, we're going to be flying all night." That wasn't one of my best jokes, but it makes fun of deans. Okay. All right. I don't have to explain it to anybody, I hope. All right. So that's what's happening in the metabolism of pyrimidines. Okay. Pyrimidine metabolism is not as interesting, I think, as purine metabolism, except for the regulation. The regulation is the key to understanding this. So the regulation of pyrimidine metabolism tells us an awful lot about how the balance of nucleotides is achieved and how that balance is important. Okay? Let me just spend a second talking about the breakdown of purines, going back to purines, because I talked about the synthesis of purines. Now I'm going to talk about the breakdown of purines. And my only reason for showing you this is to remind you of the fact that purines get broken down into uric acid. Okay? Gout, when it was first described back in the Middle Ages, was described as the rich man's disease. Because the only people who got gout were those who had diets that were rich in purines, which included a lot of red meat, a lot of red wine. No, not the red, isn't why you get it, but those two things tended to be more rich in purines, excuse me, and they would accumulate in people, especially if they weren't getting exercise and weren't being healthy and so on and so forth. So rich man's disease was gout because the diets of those people contributed to their disease. Many of the average people did not have the same access to red meat and to wines and so forth that the poorer people had. Okay. Pyrimidines don't really go to anything very exciting in terms of things that causes pain or agony or things like that. So I'm just putting this up here to convince you that there's no uric acid that's appearing in this cycle. Okay. The last thing I want to talk about is down here, deoxyribonucleotide synthesis. Everything I've told you so far indicates how we synthesize purines that are ribonucleotides, and how we synthesize pyrimidines that are ribonucleotides, and how we break down purines that are ribonucleotides, and how we break down pyrimidines that are ribonucleotides. Deoxyribonucleotides, okay, are another dimension, and they're synthesized in an odd fashion. They're synthesized from the ribonucleotides, and they're synthesized from the diphosphates. Not the triphosphates, but the diphosphates. Okay? The enzyme that converts a ribonucleotide into a deoxyribonucleotide is called ribonucleotide reductase. Ribonucleotide reductase. What does it catalyze? All right? It catalyzes the conversion of a ribonucleoside diphosphate, shown down here in the lower left, into a deoxyribonucleoside diphosphate, shown in the lower right. The difference between those two is an oxygen. That oxygen right there is taken away, and you end up with a hydrogen. We're not going to worry about the mechanism, which is what all this business is up here. If I start with GDP, I will end up with DGDP. If I start with UDP, I will end up with DUDP. If I start with CDP, I will end up with DCDP. And if I start out with ADP, I will end up with DADP, the D meaning deoxy. Okay? I still haven't said anything about T's. And it turns out that thymine nucleotides are made from the uracil deoxynucleotides. So in order for me to make thymine nucleotides, I have to first of all make DUDP. In order to make the thymine-containing nucleotides, I first have to make DUDP, which means I can't make T unless I have ribonucleotide reductase making the DUDP. Okay? Now, that turns out to be interesting and important. Okay? DUDP is ultimately made into DUMP, which is very easy to remember because if you take a dump, well, you get the idea. All right? Nucleotide metabolism is like taking a dump, right? DUDP is converted into DUMP. And DUMP is converted into DTMP. How? By putting a methyl group. That little methyl group right there is what makes it a T. That's the only difference between those two guys. And no, you don't need to know that structure. But you should know that DTMP is made from DUMP. Okay? It turns out that this guy right here that does this is that makes this uses a folate. It uses a folate. Okay? The folate is the source of the methyl group. Folates are involved in what's called one-carbon metabolism, where they put one carbon onto things. All right? Why do I tell you that? Okay? Well, I tell you that because there's the reaction. DUMP going to DTMP. Okay? DUMP going to DTMP. The folate is shown below. There's the carbon right there, and there's the carbon right there. You don't need to know the structure, all right? But that leaves behind a folate that's missing that carbon. You don't see that carbon, whereas you saw that carbon right there. Who cares? Well, you cares because you have a limited amount of folate. Just like you had a limited amount of NAD, you had to regenerate NAD from NADH, so too do you have to regenerate this folate. So you've got to remake this thing. Well, that's what this cycle is showing you. And it's not the cycle or the names or any of that I care about. But there's one thing in this cycle that I do want you to care about and know. And it's this enzyme right here, dihydrofolate reductase. You can call it DHFR. That enzyme is really worth knowing because this enzyme is one of several enzymes that's necessary to make this. But this enzyme, DHFR, is a target of a compound called methotrexate. Now, I don't have another CD to give out, but if anybody knows what methotrexate is used for, I will get you a CD from my office. Back there. It actually is used for rheumatoid arthritis. And come see me in my office, and I will. Do you take it? Do you know somebody who takes it? Do you know somebody who takes that? Okay. That's cheating, but that's okay. Yeah. Okay. Now, it's used, but that's not its primary use. The reason that I'm telling you about this here is because it's used for chemotherapy. Methotrexate inhibits this enzyme. And when it inhibits this enzyme, the cell can't do this cycle. And because it can't do this cycle, it can't make this folate. And if it can't make this folate, the cell can't make thymine nucleotides. Well, a cancer cell that can't make thymine nucleotides isn't a cancer cell. Because if it can't make thymine nucleotides, it can't make DNA. The downside, of course, being that a regular cell that can't make nucleotides also can't make DNA. So when people take methotrexate, and there are many chemotherapeutic drugs, but when people take methotrexate, they're given methotrexate for a short period of time, and then they basically flush it out of the body. And in that short period of time, cancer cells, which are dividing more rapidly, will be more susceptible and will die quicker than will regular cells. So people having chemotherapy lose their hair, get sick, and so on and so forth, because many normal cells are killed in the process. But hopefully, more cancer cells are killed by that. So this chemotherapeutic agent is targeting that guy right there. Everybody got it? Questions? There's my 30 seconds. The sooner I stop this silence, the sooner we can get done. Okay. Well, this is the last lecture of the term. And as I said, I had a kind of a boring song. It's called, if you want to see it, it's on my website. In fact, I'll put a link to my website if you don't have that already on the schedule page. They call the stuff urea, so that'll tell you all about the urea cycle. But how many people here are graduating? How many of you are going through graduation? Okay. So I'll see you there. I'm a marshal in graduation. I always like to do the graduation because it's a lot of fun. On the day of graduation, a couple things will happen. One is you're actually going to get your diploma on that day, which is OSU is really unusual and I think wonderful for doing. That's one of the reasons I participate in graduation every year, is I like to be a part of that process. We're one of the few universities in the entire country that students actually get their diploma on that day, assuming they've met the requirements for the thing. The second thing that's going to happen, besides a lot of talking and a bunch of shaking hands and so on and so forth, is everybody at the end of that is going to sing the OSU alma mater. How many of you know the words to the OSU alma mater? One? Okay. Well, as you guys know, I like to write songs, so I have an alternative version of the OSU alma mater. So if you've never learned the OSU alma mater, you can learn it here. Okay? This way, when your graduation comes, you can sing my version instead of their version. Okay? And it's actually in the form of a YouTube video. Ugly guy. Who is that guy? All right, guys. Thank you. It has been a wonderful term. I know I'll see you on finals, but I wanted to say that. It's been a wonderful term. I really enjoyed working with you guys. You have been a very patient group, and I think given what's all happened this term, I very much thank you for that, and I very much respect that. It's not easy to come in the middle of a class and work with people, and you guys have been outstanding in that respect. So I thank you very, very much for that. And I've gotten to know many of you, and I really have enjoyed that. So I wish you success, and I hope after class is done, you'll still stay in touch.